Definition on objective of medical genetics. Medical genetics is a section of general genetics that studies hereditary human disease. Disease associated with changes in genetic information called hereditary disease that have arisen at different stages of phyllo and endogenesis due to mutations under the influence of various endo- and exogenous causes. The term medical genetics has been variously defined as the science of human biological variation as it relates to health and disease. The study of the etiology, pathogenesis, and natural history of disease and disorders that are, at least, partially genetic in origin. Medical genetics initially from human. At the first, human genetics consisted largely of research and only a modest clinical component. The founders of the science are Arno Matulski, Victor McCusick, James Neal, Kurt Hirschhorn. They were internists by training. However, with the progress in medical genetics that was made possible by the conceptual and technological advances of the 1950s and 60s, particularly in the cytogenetics and biochemical genetics, and with the growth of dysmorphology in the 1960s and 70s, medical genetics became more clinical and more pediatric. Medical genetics problems. Diagnostic of hereditary diseases, analysis of their prevalence in different population and ethnic groups, medical genetic counseling, prevention of hereditary disease, prenatal diagnosis, study of molecular genetic basis of the occurrence and formation of hereditary disease, identification of risk factors of multifactorial disease. Medical genetics problems. The first, diagnostic of hereditary diseases. Next, analysis of their prevalence in different population and ethnic groups. Next, medical genetic counseling. Next, prevention of hereditary disease, for example, prenatal diagnosis. Next, study of the molecular genetic basis of the occurrence and formation of hereditary disease. And next, identification of risk factor for multifactorial disease. To a culturally heterogeneous population, India is also a melting pot of genetic diversity. The population architecture characterized by multiple endogamous groups with specific marriage patterns, including the widely prevalent practice of consanguinity, not only makes the Indian population distinct from rest of the world, but also provides a unique advantage and niche to understand genetic disease. Centuries of genetic isolation of population groups have amplified the founder effect, contributing to high prevalence of recessive alleles, which translates into genetic disease, including rare genetic disease in India. Rare diseases contribute to a significant disease burden in India. Genomics-based solutions can enable accelerated diagnosis and management of rare diseases. Biology is being transformed by the advent of technology that allows us to define the molecular basis of genetic variation. Biologists have sequenced the genomes of organisms, altered the sequence in even more, and cataloged millions of the DNA variants found in humans. The technological capacity to read and manipulate genes. We are in the early stages in this new era. Great success has been achieved with the rare monogenetic disorders for the common chronic illness that account for most of the death and disability in our society. One of example we can see in this slide. This is global distribution of hemoglobin disorders in the map of the world. Genetic counseling gives information about how genetic condition might present in family. The genetic counselor will collect personal and family health history. This information to determine how likely it is family member has a genetic condition. There are different stages in life when person might be referred for genetic counseling. There are planning for pregnancy, during pregnancy, caring for children, and managing health of adult person. In this slide, you can see the modern program of prenatal diagnosis. The screening for infection disease as hepatitis B, HIV, and syphilis, screening for inherited conditions, sickle cell, thalassemia, and other hemoglobin disorders, screening for Down syndrome, Edwards syndrome, and Patau syndrome, and screening for 11 physical conditions. This screening will be at 20 weeks of scan. We will speak about this in next part of our lecture. Genetics play a role to a greater extent in all disease. Variation in our DNA and difference in how the DNA functions, alone or in combinations, alongside the environment, contribute to disease processes. Medical genetics problems studies genetic basis of human disease, including single gene disorders, chromosomal imbalances, epigenetics, cancer, and complex disorders, and understanding and technological advance can be applied to provision of appropriate diagnosis, management, and therapy for patients. In future, you will know about many factors, environmental, genetic, and epigenetic interaction, which action together and increase risk for multifactorial disease. Better understanding of the contributions of genetic predisposition and epigenetic change to common disease, and the role of genetic alterations in response to therapy means that genetic will be relevant to everyone in the population. In second part of our lecture, we will speak about method of medical genetics. This is clinical and syndromological, morphological, biochemical, dermatoglyphics, clinical and genealogical, drawing up a pedigree, twin, cytogenetic or karyotyping, molecular diagnostic, DNA diagnostics, and population statistical method. One of the first medical genetics methods it is genealogical research, and Francis Galton is the author of the many methods for study of human genetics and genealogical method too. Slide you can see the genetic symbols for pedigree. This slide you can see one of example of Russian queen family. The next slide you can see basic terms. This is important terms which we use to our class. A proband is a patient of a carrier of a certain trait, the inheritance of which must be studied. That is, a person in relation to whom a pedigree is being built. Sips are children of the parental couple, brothers and sisters. Inbreeding, closely related crossing. The analysis includes the following steps. The first, establish whether the disease is a single one in the family or there are several cases. Family in nature. If a trait occurs several times among generations, then we can assume that it has a hereditary nature. The second, determine the type of inheritance of the trait. Next, compilation of a pedigree begins with a proband. A brief record of data about each member of the genus is kept, indicating its relationship to the proband. Next, genealogical data should be obtained from at least 3 generations of ascending and lateral relatives about all family members, including grandchildren. Next, enter information about miscarriages, abortion, stillborn children, infertile marriages, and so on, including early deaths. The next, the age of the pathology manifestation must be indicated. And the last, the more generation traced in the pedigree, the more complete is this, and, accordingly, the higher the chance of obtaining reliable results. Genealogical method help to us understand about disease in family. It is dominant or recessive, X-linked or Y-linked. What we will analyze it in ready pedigree. At first, parents' illness, presence or absent, disease in woman more or in men, incidence in generation. It means vertical distribution of the disease. The likelihood of having a sick child, incidence among siblings, or horizontal distribution of the disease. Which character in autosomal dominant inheritance? The pathological sign is found in every generation of the pedigree. The trait manifests itself in homozygous and heterozygous gene carriers. If there are many siblings in a given generation, then the ratio of sick to healthy siblings is close to. Both sexes are affected in equal proportions. Father and mother pass the mutant gene equally to daughters and sons. Transmission of the disease from father to son is possible. The manifestation of disease may not develop immediately after birth. The penetrance of pathological manifestation is not always 100%. This is due to the fact that heterozygotes can occur in generations, but without pronounced sign of the disease. Heterozygosity is evidenced by the appearance of the disease in some of their children. Different severity of clinical manifestation. Clinical manifestation of the disease may vary depending on the expressivity and penetrance of the mutant gene. The dominant gene can have varying degrees of the expressiveness in severity which makes it difficult to establish the type of inheritance. You can see pedigree of dominant autosomal disease, and you can see vertical and horizontal and not independent from male and female. And next slide you can see example of autosomal dominant disease. Which character we know about autosomal recessive inheritance? It is the disease is rare in the pedigree and not in all generations. The risk of having a sick child is only 25%. The probability of illness in girls and boys is the same. The trait can manifest itself in children whose parents were healthy, as they were heterozygous carriers of the mutant gene. A mutant gene manifests its action only in a homozygous state. In a heterozygous state, it can exist for many generations without manifestation itself phenotypically. The incidence of hereditary autosomal recessive disease is in direct proportion to the prevalence of the mutant gene among the population. It increases in isolation populations and in families with a high percentage of closely related marriages. And last, different degrees of expressiveness and penetrance of the trait are possible. Autosomal recessive disease include phenylketonuria, galactosemia, albinism. Diseases are usually diagnosed at an early age. This slide you can see one of example of autosomal recessive disease, Harlil Profounder-Guiner disease, or gargolinism. Next, X-linked dominant inheritance. The phenotypic manifestation of the disease will have both homozygous and heterozygous. In healthy fathers, sons and their children will be healthy, since only the Y chromosomes can be passed one from the father. Daughters of homo and heterozygous will be phenotypically sick. The literature describes pedigree for some diseases with this type of transmission, which do not have male siblings, since a strong degree of damage causes their death in utero. In this slide, you can see character sign of dominant X-linked disease. And as example, we know about phosphate X-linked recessive inheritance. Sons will be sick, since they have only one X chromosomes, and all recessive and dominant all times will be as dominant. The statistical ratio of healthy and sick men is 1 to 1, and example of this disease hemophilia, Duchenne myopathy. In this and the next slide you can see example of X-linked recessive disease. It is hemophilia with blooding and color blindness, when people cannot make difference between red, green, and other color. Color blindness or daltonism. John Dalton, it is first who description of the defect he had discovered in his own and his brother's vision. Y-linking inheritance type. Passed only to sons. With the Y chromosomes, such signs are inherited as hypertrichosis, the presence of hair along the edge of the auricles, skin membranes between the fingers, development of the testes, the intensity of growth of the body, limbs and teeth. Cytogenetic research. Diagnosis of chromosomal disease is carried out using cytogenetic analysis. The material for cytogenetic studies in human cells, practically any tissue and organs at any stage of the cell cycle, in mitosis and meiosis, peripheral blood lymphocytes, bone marrow cells, fibroblast, tumor cells, and embryonic tissue. Indication for the study. The woman's age at the time of delivery is 35 years or more. Suspected chromosomal disease. Multiple congenital malformations in children already born. Several cases of spontaneous abortions, stillbirths, congenital malformation in children, and a significant delay in the mental and physical development of the child. Reproductive dysfunction. There are direct and indirect methods for studying chromosomes. At the first, direct methods are used in the study of tissue with high mitotic activity, for example, bone marrow, lymph node cells, embryonic tissue at early stage of development, and chorion, or a placenta, and any stage of pregnancy, and in the study of meiotic chromosomes. Second, indirect methods include obtaining preparation of chromosomes from any tissue after stimulating cell proliferation in culture conditions for a period of time from several hours short-term cultures to several years continuous cultures. In clinical cytogenetics, methods of analysis of buccal epithelium and peripheral blood lymphocytes are widely used. In this slide we can see research of number chromosomes, and in left and right picture we can see both 2 and 2 blue spots. It means we use control and we can see diploid. But in left picture we can see only one spot, chromosome number 6, and we can speak about monozyemia. But in right picture we can see tree spot. It means trizymia of chromosomes number 6. The next method it is twin method, proposed by Galton in 1875 to differentiate the role of heredity and environment in the development of various traits in humans. The essence of the method is to compare the manifestation of a trait in different groups of twins. The frequency of occurrence of traits and difference in their genotypes are taken into account. There are two groups of twins, monozygous and dizygotic. Dizygotic twins result from the simultaneous fertilization of two distinct eggs by two different sperm. Dizygotic twins have about 50% of genes in common and can be either same sex or opposite sex. The result of comparing these two groups of twins in the calculation of indicators of compliance or concordance and inconsistency or discordance, as well as the calculation of the frequency of occurrence of the trait in each group of twins. Concordance, an indicator of the identity of a pair of twins on a certain basis, corresponds to the proportion of similar pairs for the studied trait among the survived pairs of twins for each group. And in slide you can see formula for concordance, monozygotic and dizygotic. Discordance is an indicator that is calculated for dizygotic twins as the proportion of discordant twins. The trait was detected only in one twin out of a pair, according to the trait under study among all survey dizygotic twins. And for find discordance, we use formula which present in this slide. In this slide you can see two brothers, dizygotics, and different habitus in each of their pair. The next method it is population statistical. Studying hereditary traits is large group of the population in one or more generations. Using statistical processing of the obtained data, the method can be used to calculate the frequency of occurrence in population of alleles of a gene and the corresponding genotypes for these alleles. To find out the distribution of various hereditary traits in it, including disease. To study the mutational process, the role of heredity, and the environment in the formation of human phenotypic polymorphism according to normal signs, as well as in the occurrence of disease, especially with hereditary predisposition. The basis for elucidating the genetic structure of a population in the Hardy-Weinberg law of genetic equilibrium reflects the pattern according to which, under certain conditions, the ratio of the alleles of genes and genotypes in the gene pool of a population remains unchanged in a number of generations. The mathematical expression of the Hardy-Weinberg law is this formula: P big A plus Q small a, all this in square and equivalent 1. In this formula, we use P and Q as frequencies of the A big and A small or dominant and recessive alleles of the corresponding gene. An analysis of the frequencies of occurrence of different traits in a population if they comply with Hardy-Weinberg law suggests that traits are due to different alleles of the same gene. The next biochemical research, based on the fact that with mutations of enzymes and transport proteins, they can be detected by the accumulation of pathological metabolites, substrates of enzymatic reactions, or substances transported through the membrane, amino acids, sugars, glucosaminoglycans, keto acids, accumulate in urine and blood serum. For example, with a lack of phenylalanine hydroxylase, phenylalanine and its transaminations products, phenylpyruvic and phenylacetic acid accumulate in the blood and tissue. Both acids are toxic to the baby's brain. You can see indication for biochemical studies. There are unusual hair, nails, face, mental retardation, hyperreflexia, convulsive syndrome, defects of behavior, motor disinhibitions, muscle hypotonia, violation of speech, organs of vision, hearing, anomalies in the development of the skeleton of non-rickety nature, chronic ingestion of a non-infectious nature, unclear and protracted forms in children during the neonatal period in the first years of life. These type of methods are used when you suspect hereditary metabolic disease. They can be multi-stage or screening or immediately strictly directed to a specific pathology. In the practice of healthcare in the Russian Federation, a two-stage program is used. The first, screening program, mass and selective screening, and the second step, methods to confirm the diagnosis. In the country, diagnostic phenylketonuria, congenital hypothyroidism, adrenogenital syndrome, congenital malformation of the neural tube, and Down disease are carried out. Molecular genetic study allows you to diagnose this hereditary disease at the level of genetic defects. Direct diagnosis of mutation includes several methods: determination of the nucleotide sequences in the genes of their fragments, detection of violation of the restriction site, allele-specific hybridization with synthetic oligonucleotide probes, chemical and enzymatic cleavage of DNA at sites in incorrect base cross-linking, registration of change in the electrophoretic mobility of mutant DNA molecules, in vitro translation in the protein product, nucleotide sequence method. Immunogenetic study. The method allows identifying hereditary primary immunodeficiencies, the compatibility of the mothers and the fetus, and determine the prognosis in the disease with a hereditary predisposition. The following so-called warning signs can serve as indication for conducting. Frequently disease of otitis media, at least 6 or 8 times during 1 year. Several confirmed serious sinusitis, at least 4-6 times within 1 year. More than 2 confirmed pneumonia in 1 year. Recurrent deep abscesses of the skin or internal organs. The need for long-term antibiotic therapy to stop the infection, up to 2 months or more. The need for intravenous administrations of an antibiotic to stop the infection. At least 2 deep infections, such as meningitis, osteomyelite, sepsis, lagging behind the infant in height or weight. Presence of primary immunodeficiencies in the family. Facts of early death from severe infection or the presence of one of the listed symptoms. Persistent trace of fungal infection of the skin over the age of 1 year. The study of the immune status is immunodeficiency should include the study of the amount and functional activity of the main components of the immune system. For example, phagocytic system, complement system, T and B immune system. The methods used to assess the function of these systems are conditionally divided into tests of the first and second levels. The first tests are indicative and aimed at detecting growth defects in the immune system. The second tests are functional and are aimed at detecting a specific breakdown in the immune system. And our last part, the third part of our lecture, it is principles of prenatal diagnosis. Diagnosis of hereditary disease is primary and secondary, the essence of which is to prevent the birth of a sick child. Areas of primary diagnosis include the first, family planning, prospective or before the birth of a child, and retrospective, counseling, or after the birth of a sick child. And the second, prenatal diagnostics, with the elimination of pathological embryos and fetuses. Secondary diagnosis provides for the correction of the disease after birth, normocopy, the formation of a healthy phenotype with a pathological genotype. In this slide, you can see indication for prenatal diagnosis. There are: the age of woman is over 35-37 years old. Men are over 45 years old. The birth a child with a chromosomal of gene disease. Presence in the family of a well-established hereditary disease. Carriage by the mother of the X-linked recessive disease gene. A history of spontaneous abortions in the early pregnancy, stillbirth, birth of children with developmental defects and chromosomal abnormalities in pregnant women. Pregnant women from the zone of high radiation background. The presence of the consanguineous marriages. The presence of the similar cases of the disease in the family and among distant blood relatives. Recurrent chronic long-term treatment-resistant course of the disease. The presence of rare specific symptoms of the combination. Multiple damage to organs and organ systems. The presence of more than 5 congenital morphogenetic variants, also called development microanomalies, congenital nature of the disease. The prenatal diagnosis may be indirect and direct methods used. With indirect methods, a pregnant woman is examined, for example, a blood serum test for alpha-fetoprotein. With direct methods, the fetus is examined. The invasive ones include pre-implantation diagnostics on the 1-2 cells taken at the early stages of cleavage from an in vitro fertilized egg, 8 till 18 cells. Chorion biopsy taking the epithelium of the chorionic villi. Placenta biopsy, amniocentesis, obtaining amniotic fluid and fetal cells. Chordocentesis, taking blood from the umbilical cord. Biopsy of the skin, liver, muscles of the fetus. Vesicle synthesis, puncture of the fetal bladder wall to obtain urine. Fetoscopy, examination of the fetus with a fiber optic endoscope inserted into the amniotic cavity through the abdominal wall of the uterus. The method allows you to examine the fetus. Umbilical cord, placenta. Non-invasive ones include ultrasound examination, screening study of the blood serum of a pregnant woman, determination of alpha-fetoprotein, chorionic gonadotropin, and other. We can speak about example of hereditary disease. And the first, I give two examples of monogenetic disease. Diseases associated with gene mutations can be monogenetic when one defective gene became cause of one disease. And this example, phenylketonuria, an autosomal recessive disease caused by a metabolic disorder of the essential amino acid phenylalanine, which enters the human body with protein food. We can speak about some types of phenylketonuria. It may be type number 1, phenylalanine 4 hydroxylase deficiency, and phenylketonuria type 2 and type 3 when defect happens with pterin cofactor. These cases meet in clinical practice more or less than phenylketonuria type 1. In this slide, you can see a distribution of phenylketonuria disease. And we can see that incidence disease children for 100,000 population in many countries. In this disease, need understand phenylketonuria. It is autosomal recessive disease. In different countries, distribution will be very differences. For example, more often it is in Turkey, Ireland, Scotland, Israel, Czech Republic, Brazil, Australia. More seldom it is Finland, Japan, Philippines, South Korea. In this picture you can find India. I marks your country in red, and it is middle than other country. And if we will speak about Russia, in Russia, in different towns we have different statistical dates. For example, in St. Petersburg, it may be 1 to 7,600 In Moscow, it may be 1 to 67 born persons. In this slide, we will speak about how mother and father can transmit disease, and in which cases it cannot happen. For example, you can see at figure 1. You can see that mother and father both has recessive genes of disease of phenylketonuria. So 3 parts of healthy, one of them absolutely healthy, 2 heterozygotes, and 1 part will be with disease, homozygotes of recessive disease gene. In figure number 2, we can see how both parents has disease genes, recessive genes, but their children not have disease. How it happen? If you careful to see in this picture, you will look. Mother have phenylketonuria disease gene, type number 1 or mutation in position number, for example, first. And father has disease genes too, but it will be mutation other type. For example, gene mutation in position number second or third. So, in their children will combination with healthy and disease genes, but every disease genes, recessive genes, will cover dominant healthy genes, and this is child not has disease but has two defective phenylketonuria recessive genes. As a result, in every population, phenylketonuria disease genes can meet one carrier of disease gene in every 50 healthy persons. But when happens meet between woman and man, and meet between different type of mutation genes, it not always leads to disease. If we again return to our disease with distribution phenylketonuria in different country, we will can see more distribution this is disease has when population close, and many family has couples between cousin and cousin, and disease meet seldom when this is marriage not happens in country or culture or populations. Previously I said about different types, 3 types of phenylketonuria. In this slide you can see description or explanation of every type. For example, type first, deficiency of the enzyme phenylalanine hydroxylase leads to the accumulation of phenylalanine and its decay production in biological fluids. And you can see position in chromosomes and localization on long or short arms of chromosomes. It means disease type number 2, deficiency of cytosolic dehydropterin reductase, and defect in position 4p15.3. How we can understand about this disease? This is disease at first begin in after birth, maybe second till 6 months in the first years of life. It will be lethargy, lack of interest in the environment, sometimes increased irritability, regurgitation, hypotension, convulsions, dermatitis phenomena. Then delayed muscle and mental development, microcephalus and hydrocephalus, hypopigmentation of the skin, epileptic cases. The course of disease will progressive, and if not use treatment, it will be cause of death, and age of child will be 2 or 3 years of age. So it's very important. Very important to know present disease or not in a newborn child. In many countries, present screening program to everybody after born for first day of living. Take blood and send in genetic laboratory. This is blank with spot of blood. You can see in right, because if parents will know about disease of their child, it can use treatment many days early. The main treatment in this case will include not give food consist of phenylalanine, and it is many difficult because many food consist of phenylalanine. The next example, sickle cell anemia. Mutation in the beta subunit of hemoglobin gene. It is chromosomes number 11 and short P arm. Defective hemoglobin is formed by replacing valine with glutamic acid. Function of giving oxygen to tissue, hemoglobin polymerases with the formation of fibers that deform erythrocytes with the formation of long chains. The erythrocytes became sickle-shaped. The patient with this disease will has increase in blood viscosity, stasis, a mechanical barrier is created in small arterioles and capillaries, which leads to tissue ischemia. In more often, it will associated with pain crisis, and anemia will progressive during physical activity. The cause of this disease more often missense mutation, when the replacement of a pair of nucleotides led to the replacement of an amino acid in the protein, or the primary structure will change. This is reason of change secondary, third, and fourth shape of erythrocytes. And shape of this erythrocytes you can see in left photos. It normal round shape, and in right part of the slide you can see sickle cell of erythrocytes. In this slide you can see character shape of erythrocyte. It is lower line of photos, and in upper part of slides you can see character clinical picture or habitus of patients. For example, if you can see for this little boy, you will see his head is very long, long as column. It will be show that his tissue all times when little organism was development has hypoxia, and tissue formed with this character. In the next picture you can see hand with different long of fingers and shape as a watch glasses. It is character for hypoxia too. But beside this missense mutation, the cause of disease may be nonsense mutation. It means when nucleotide substitution and as a result of a shift in the reading frame. Example of this mutation will be blood group 0. In people with this blood group, a single nucleotide was lost or deleted in the gene. In this slide you will see scheme when antigens A and B are oligosaccharides synthesized from antigen H under the action of enzyme proteins A or B. Zero mutation in the gene led to the formation of an active protein. Next, very sad and very interesting disease, it is progeria. This is a genetic disorder in which senescence appears to be gently accelerated. In this photo you can see three persons which have next age from left to right: 15 years old, 12 years old, and 26 years old. If we will describe this disease, we can say about this is very interesting habitus. This is patients looks as a children, but if we will speak about their skin, we understand they have looks as old people, but it is not correct if we will speak about absolutely old because we can see the bone not grows and shape and size of head, leg, arm and all skeleton as a child. The next example, it is neurofibromatosis type 1. It is disease autosomal dominant. It is associated with tumor. This is disorders can affect multiple body systems. It is marked by skin changes such as scare outlet spots, irish ridge nodules, which are begin growth on the colored part of the eyes, skeletal abnormalities like scoliosis, large head size, and learning disabilities. And here I can say about many other tumors have somatic mutation in their cause, and many specific genes involved in the control of cell division, differentiation, proliferation, and death of cells. So it may be a cause of the development of many oncological diseases. All oncogenes may be dominant or recessive. If we will speak about dominant oncogenes, so in anamnesis of patients, we all times will have disease in from every or more part of members of family, But if we will speak about recessive oncogenes, so it may be sporadic case in family, and it may be after action different risk factors for people, and only then leads to oncological disease. The main difficulty in understanding the genetic basis of multifactorial disease, which include most common human disease, including arterial hypertension, coronary atherosclerosis, heart failure, diabetes, and so on. And if we will speak about differences between monogenetic disease and multifactorial disease, we need to speak about next. Monogenetic disease. What leads to this type of disease? Mutations. If we speak about multifactorial, polymorphism. It is maybe mutations, but it is changes in genome or DNA molecule, but it is not abnormality. It is a variation as a result in protein, and so it is if we find one polymorphism genes in DNA, not leads to disease, but in some with environmental factors, in intergenetic interaction, and common action of different polymorphism will lead to multifactorial disease. Next example of hereditary disease, it is a disease which connection with chromosomal aberration or chromosomal disorders. And here we can speak about disruption of chromosome structure, interchromosomal, isochromosomal. And in slide you can see many differences between abnormality chromosomes. And if we speak about Fiji 1, chromosomes number 1, it is normal. Then example of deletion. The third example of duplication. The fourth example of transversion, and example of trisomia. All these type of disorders may be in interchromosomal, Next, what about we will speak? It is information about the most mutations, chromosomal mutation, are lethal, and it is way of safe normal karyotype, human karyotype, and karyotype of other animals for new and new generation, and elimination new variant of chromosomal. But sometimes in karyotype can meet the damage or disruption character not for all cells in all organism. This is situation if mutations occur during organogenesis and not take part in all cells, we will call as mosaicism. In this slide, you can see two mechanisms how it may be appearance. Let's to see in right side of slide. Trisomia correction. We can see at first was trisomia, and organism and all cells in this organism has 47 chromosomes. But then happens mutation. For example, during mitosis, one chromosomes cannot move and stay in with your pair chromosomes. As a result, one part of cells has 46 number of chromosomes in cells, but other has 48. This is example we will call as non-divergence. And this is we can see that mosaicism leads to three type of cell clones: 47, 46 normal, and 48. The other example, anaphase leg, it is only two type of cells, but part of them present abnormal amount of chromosomes 47 and part present as a normal karyotype. The next example in right part of slide, we will speak about mitotic errors or euploid zygote. And here situation was opposite. When normal karyotype, normal amount cell chromosomes leads to abnormality, and non-divergence when in mitotic error happens three type of karyotype in one organism: 46 more part of cells, normal karyotype, and less part, 45 chromosomes in one type of cells, and 47 in other type of cells. And anaphase leg, opposite condition, when only two types cells in one organism, more cells have 46 chromosomes in each other cells, and less amount of cells have 45 abnormality amount of chromosomes. The next type of chromosomal aberration or errors, we will speak about type bilocalization in the chromosome. And here we can speak about the first, autosomal, in autosomes or non-sex chromosomes. And the second type, it is linked with X chromosomes or linked with Y chromosomes. Next part of our example, it is genomic. And here we will speak about some types. For example, when from every pairs stay only one chromosomes. For example, X0, monosamia, or polysomia when we can see in karyotype more than 2, for example 3, 4 or 5 chromosomes from one pair. It may be trisomia as a Down syndrome. And the next type of genomic examples, it is aneuploidia and polyploidia when we speak about karyotype which has haploid or triploid or fourploid disorders. Some of sex chromosomal abnormalities examples you can see in this slide. For example, it is Klinefelter syndrome, trisomia X, Shereshevsky-Turner syndrome. And now we will speak about every of these syndromes. Look at this picture, and we can see Down syndrome. It is genotype 47, XX or XY, or man, woman or man, 21 plus, or trisomia for 21 chromosomes. And in picture you can see my notes in 21 pairs. Patients with Down syndrome has excellent for remember habitus. It is always same habitus for different people populations. And if you see first time Down syndrome phase, you always can recognize in your patients this is type of disease. It is wide face, tongue, big. In hand you can see a line. In eyes you can see as a bird's epicant. Chinese type of eyes, and side of eyes, inner side little down. Size or distance between two eyes wide 2. Hand short and wide, and last finger very short, and sometimes may be torn in inner. Sometimes about these children with disease Down syndrome say as son children, because it is very kind children. The next, Turner syndrome, and it is genotype 45X0. And in picture you can see female phenotype, height very small, for example 145 centimeters, and underdeveloped secondary sexual characteristics. The next, it is Shereshevsky-Turner syndrome. Many newborn girls show only minor symptoms, but some already have deformities of their hands and feet, as well as folds of skin on the back of their necks. Other symptoms of this syndrome include a specific facial expression. Facial expressions are weak, the inability to wrinkle one's forehead, the mouth is always half open, the lower lip sags slightly. The shape of the chest looks like a burial. Swelling of the lower extremities. Low growth is combined with high body mass. The hairline on the neck is low. Nipples are localized much lower and wider than in healthy children. Significant puberty disorders can be seen in adolescents. Lack of periods, hairline on the pubis and in the armpits, underdevelopment of the mammillary glands. Stunting. The older the child becomes, the brighter this symptom appears. Coverture of the dentition, observed with all genetic disease. Strabismus, mental retardation. Children are characterized by carelessness and poor memory. Mental disorders in the form of emotional instability, depressive states, psychosis, constant anxiety, malformation in internal organs, the cardiovascular system, hearing organs, and kidneys. Their doubling is often observed. Facts. The syndrome was named after the scientist who made the greatest contribution to the study of this pathology. In 1925, the Soviet endocrinologist Shereshevsky suspected the hereditary nature of the disease. Later, in 1938, the American physician Turner described the most characteristic symptoms. In English literature, the disease is sometimes referred to as Aldrich-Turner syndrome, referring to another researcher. Chromosomal disorders, single-sex chromosomes X, which caused the development of Shereshevsky-Turner syndrome, was discovered only in 1959 by scientist Ford. The pathology is unique in its kind. The fact is that children are viable only in the absence of a second sex chromosome. If any other chromosome is absent, then the fetus, after conception, does not develop at all, or development begins, but the child dies in the prenatal period. Thus, Shereshevsky-Turner syndrome is the only option in which a woman lives with 45 chromosomes. The next example, Klinefelter syndrome. The main signs and syndromes. Small secondary signs of sex signs, diminished pubic, ciliary, reduced facial and body hair, sexual dysfunction, enlarged breast tissue, tall stature, abnormal body proportions, learning disabilities, personality impairment, a single crease in the palm. The next example of hereditary disease, it is dependent by localization in the cage. It may be DNA which meet in nuclear and cytoplasmic. we will speak about mitochondrial disease. Mitochondrial disease are maternally transmitted and affected muscles, vision, and the nervous system. It is congenital malformations and dependence from period of prenatal development. We can divide it all this period for three steps: blastopathies, embryopathies, and fetopathies. Period, if we speak about days or weeks, we can see in this table blastopathies during 1 till 15 days after fertilization. Embryopathies from 16 days till 8 weeks. Fetopathies, we speak about period from 9 weeks before birth. In this slide you can see how often different forms of hereditary pathology frequently per 1 or meet per 1,000 person. For example, if we speak about monogenetic, in common it may be 5 to 15 for every 1,000 person, and includes autosomal dominant from 2 before 10 newborn for every 1,000 newborn persons, autosomal recessive 2, 4, and X-linked from 0 before 15 newborn, frequently per 1,000 newborn children. Chromosome disease can meet 5, 6, 7 person for every per 1,000 persons newborn. Multifactorial, it is 7 till 10 for every 1,000 persons. And congenital malformations, it more often, because it is not disease and it is malformation with more often to meet in newborn, and in statistical date it means 19 from 19 before 22 type of genetic disorders or congenital malformations. It is deformities, defects of morphogenesis, the most severe manifestation of congenital malformation. Dysplasias, it is deformities, morphological congenital change that go beyond the generally accepted norms. Minor developmental anomalies, it is stigmas of dysembryogenesis as syndactyly, dimples on the cheeks, curvature of the little finger, defects that do not require medical intervention. And last, clinically significant malformations. It is congenital anomalies requiring medical attention. In this slide you can see the teratogenetic agents which can act as a factor leads to congenital malformation. For example, it may be medicines, alcohol, marijuana, heroin, cocaine, hyperthermia, substances that pollution of environmental and infection disease. This slide, a photo of child which has many congenital magnification. He has not excellent development of hands and little not excellent development of legs. It is demonstration of effects of thalidomide. What means thalidomide? It is drugs which was very famous and very popular in middle last century. Pharmaceutical company, when produced this drug, was very sure because it was drug with excellent sedative effects and made very calm all people. So it was very famous and given and taken all pregnancy woman which was very nervous. But after some times became born many children with same phenotype or habitus. So it was very interesting. This slide you can see one more classification of hereditary disease. Previously we speak with you about different type of classification hereditary disease. It was genetic, chromosomal, genomic. It was mitochondrial and nucleus. It was disease and congenital malformation. And now you can see new classification and read example of this classification of hereditary disease.